November 1983. The body of a 15-year-old girl, Linda Mann, is found in a village near Leicester.
It's still raw. Yes. And I can't believe that after 35 years. She's such a lovely girl. It was wrong.
3 years later, and a second girl is found less than a mile away. Dawn Ashworth was also 15. When Dawn's body was found, police knew straight away they were looking for a serial killer. What happened during the course of this investigation would change police detective work around the world forever. David, just explain what all this pile of stuff is. These are mementos from the case, are they?
David Baker, who's now 84, was leading the double murder investigation. He'd arrested a 17-year-old, Richard Buckland. Buckland had admitted the second murder, but there was a problem.
There was no circumstantial evidence against him on the first murder, and he totally denied it.
You were convinced it was one murderer?
Yes.
Constable Baker is certain Richard Buckland killed both Linda Mann and Dawn Ashworth. Struggling to make a case, Baker has an unusual idea.
I was aware at that time that Dr. Alec Jeffreys had been working at Leicester University in respect of DNA tests.
Dr. Jeffreys knew human DNA is mostly identical.
And well over 99% of our DNA is the same between people. That allows us to have 2 ears, 2 eyes, a nose, 4 fingers and a thumb on each hand.
But Jeffreys wanted to know more about that other 1%. In 1984, he discovered a patch of DNA that was unique to each individual, a sort of DNA fingerprint. And when Constable Baker thinks about Jeffreys' work, he has a question. If DNA can prove paternity, can it also prove whether Richard Buckland killed Dawn Ashworth and Linda Mann? The police are willing to try anything. They give the lab the crime scene samples. These researchers may know DNA, but they've never used it to solve a crime. The results of this real-world experiment are anybody's guess. First, technicians extract the DNA by dissolving the evidence. At that time, they needed a sample as large as a quarter to generate a DNA profile. Then, Jeffreys has to pinpoint the DNA fragments that are unique to their owner. To do that, he uses a technique called RFLP, Restriction Fragment Length Polymorphism.
And what this does is that it cuts the DNA into sections of various sizes.
To get the DNA portrait he needs, Jeffreys has to sort the fragments by size. Then the sample goes onto one end of a gel plate. Technicians apply electrical current, and electricity pulls the fragments through the gel.
The gel acts exactly like a sieve, and the small fragments are able to travel quickly through the gel, and the large fragments travel slowly. At the end of the process, we have an X-ray film, which effectively has a barcode on it.
It may look like random splotches, but that barcode is unique. In all the world, it belongs to only one person. And soon, the results are in.
The 2 murders, they had been committed by the same individual.
But then, the story takes an unexpected turn. The DNA portrait doesn't match that of the one and only suspect, Richard Buckland. Despite his tormented confession, Buckland didn't kill a soul.
They came back not connected. The result was stunning.
A blow to you?
Well, it was a blow because it meant that we'd got the wrong man.
Buckland was released. The very first time DNA profiling was used in a criminal case, it exonerated a suspect. David Baker was back to square one. 2 murders, 1 killer, no suspect.
It says, will the killer strike again? How can you help to trap him?
We made a decision there and then to use the DNA system as part and parcel of the inquiry.
What happened next was extraordinary. Police asked all the men who lived in the local area to give a sample of their DNA. More than 5,000 men gave a blood sample voluntarily, and eventually the killer was caught.
This is Pitchfork. He went from being a very normal family man to a murderer in a split second.
Colin Pitchfork, a local baker, was sentenced to life, the first person ever to be convicted on the basis of genetic evidence.
Today there are ongoing debates about DNA, how the information is stored, who should have access to it, how it's used. But Linda's family have no doubts about genetic profiling and the historic role it played in solving a double murder.
The Forensic Biology Unit examines evidence recovered from crime scenes for potential biological matter. They identify biological fluids, then take swabs or cuttings from the evidence and process them for analysis. Cases that test positive in the Forensic Biology Unit go on to forensic DNA analysis. The goal of the DNA unit is to develop DNA profiles from items of evidence and compare them to known DNA profiles from crime investigations. Evidence is examined from many types of cases, including homicides, sexual assaults, robberies, aggravated assaults, kidnapping, and property crimes like burglaries and theft from a motor vehicle. Cold cases are also examined, including homicides and sexual assaults dating back to the 1960s. Work in the Forensic Biology Unit begins with a visual examination of evidence for staining. Biologists take samples of potential stains to test for the presence of blood, semen, saliva, or cellular material. Hairs can also be tested if they contain roots suitable for DNA analysis. Stains not visible to the naked eye can be located with an alternative light source. Some body fluids fluoresce under this blue light, making it a useful tool for finding stains that aren't visible using natural light. Any biological evidence obtained is stored in a walk-in freezer until it's ready for DNA analysis. The DNA analysis process uses a variety of manual and cutting-edge robotic methods to analyze samples and develop DNA profiles. Every DNA sample goes through a 4-step process. The first is a DNA extraction, which is done to purify the DNA in a sample. After extraction, samples are passed through a secure chamber from the pre-amplification to the post-amplification laboratory. The next step is DNA quantitation, which determines how much DNA is present in each sample. That's followed by an amplification step, where the DNA is copied millions of times. Finally, the DNA is detected on an instrument called a genetic analyzer. This machine assembles the finished DNA profile, which analysts use to determine if biologic evidence from a crime scene matches the known DNA profile of a victim or suspect. DNA cases that are submitted without a known suspect can be searched in the Combined DNA Index System, or CODIS database. This is the largest DNA catalog in the world, with over 14 million profiles. CODIS is used by law enforcement agencies in the United States to help solve unsolved cases.
What is DNA fingerprinting or DNA profiling? Leicester University geneticist Alec Jeffreys developed a technique called DNA fingerprinting in 1985. It allows DNA samples from different people to be compared to look for similarities and differences. It's used for solving crimes and can also confirm if people are related to each other, like in paternity testing. Any two people in the world have 99.9% of their DNA the same, so this process analyzes the differences in the remaining 0.1%. This modern technology is called DNA profiling. It's a very sensitive technique which only needs a few skin cells, a hair root, or a tiny amount of blood or saliva. There are sections, or loci, of chromosomes, where instead of a gene consisting of a long sequence of bases, there are much shorter sequences of 3, 4, or 5 bases that are repeated many times. For example, these repeated sequences are called short tandem repeats, or STR. At these places on the chromosomes where we find these STRs, there are areas that vary in number of repeats. DNA profiling only looks at these STRs. A cell sample is collected. This could be from some blood at a crime scene, or a swab from the inside of someone's cheek, for example. The DNA is then extracted from the sample. Many copies of this DNA may be made using the polymerase chain reaction, or PCR. Special enzymes called restriction endonucleases are used to cut the DNA up into different size pieces. The DNA samples are then put into wells in a special gel called agarose for the process of gel electrophoresis, which separates the DNA fragments by size. The pattern is then transferred to a nylon sheet in southern blotting. And finally, the lines produced by the DNA samples from different people are compared. So let's have a go at using DNA profiles to solve some mysteries. An item was stolen in a burglary. A drop of blood was left behind by the thief. Samples of DNA were taken from 4 suspects and compared to the sample left at the crime scene. Which suspect is guilty? Here's a clue. Look for the one that is most similar to the one from the crime scene. Pause the video while you decide. Suspect 3 is guilty. Can you see how the pattern of bands matches in Suspect 3 and the crime scene? Their DNA is the same. Let's try another. A soldier has been killed in an explosion and has lost his dog tags that identify him. 3 soldiers are missing from their unit, so the army asks the 3 sets of parents for a DNA sample so that they might compare it to the soldier's DNA and therefore make an identification. Remember, the soldier will only share half of his DNA with each parent. So which set of parents is the soldier the son of? Pause the video while you decide. Parents C and D. As you can see, he inherited the first band from parent D and the second, third and fourth from parent C, and so on. So now you understand how DNA profiles or fingerprints are made and seen some examples of where they can be used. You can even interpret a DNA profile.
DNA is life's blueprint, a molecule that encodes genetic instructions used in the development of all known living organisms and viruses. It's a nucleic acid that in most cases are double-stranded helices, consisting of 2 long biopolymers made of simpler units called nucleotides. DNA is tailor-made for biological information storage, which is why it's so useful in the world of forensic science. So how is it utilized? Forensic DNA analysis is a multi-step process, starting with removing the DNA from the biological material that is submitted to a lab. This can be saliva, blood, semen, hair, skin cells, or any biological material containing cells with a nucleus. The next step is extracting the DNA from the nucleus, a process called extraction, where cells are broken open using a combination of chemicals and heat to release the contents of the nucleus where the DNA is stored. The materials then go through a cleanup process so the sample is pure DNA. The next step in the process is to determine how much DNA was retrieved from the cells through a process called quantitation. Quantitation is important because the next step requires a specific amount of DNA to achieve optimum results. Too much or too little requires the concentration to be adjusted before the next step. After quantitation comes the amplification process where specific regions inside the DNA molecule are copied. During the amplification process, fluorescent tags are placed on regions of the DNA, which will be used in the last step. The last phase is the detection process, where our copied fragments of DNA are separated by size and are passed through a laser. When this laser hits the fluorescent tags we added earlier, a camera determines what color the tag is and how much there is. What results is a DNA profile set of numbers at the different regions tested called a locus. One of the numbers comes from the mother, the other from the father, linking the subject to each one. Currently, DNA testing gives results at 16 different areas. Results at all 16 areas is considered to be a full profile. If an individual's DNA profile matches at all 16 areas to a DNA profile developed from evidence at a crime scene, then law enforcement can use that data to place the individual at the location.
Imagine this. 2 cops come to your door and arrest you for murder. You know you didn't do the crime. In fact, you were in the hospital at the time the cops say the murder was committed, and you have records to prove it. But the cops have evidence too. Your DNA. It was found on the dead man's finger. How could that have possibly happened? I've been looking into DNA and whether it's as reliable as we all think it is. We all have a lot of faith in DNA, and it is pretty conclusive. It's better than any other forensic science we have, especially when you have a large amount of one person's well-preserved DNA. To interpret DNA, analysts look at a chart called an electropherogram. A perfect sample of DNA comes out like this. But in criminal investigations, finding a perfect sample is actually not the norm. Sometimes analysts are working with degraded samples that are old or have been exposed to the elements. See how the spikes start to fade away? Or they're interpreting a sample with many people's DNA, and all the spikes combined together. It can be hard to tell whose DNA is whose. Sometimes they're working with tiny amounts of DNA, maybe a piece of evidence the suspect touched. In these cases, the spikes can be small and difficult to interpret. Some labs won't even analyze these samples, but some will, even though the margin of error is higher. Technological advances will probably solve some of these problems and make DNA analysis more accurate. But as technology advances, it will also introduce new problems. Someday soon, analysts may be able to detect the genes from a single cell, and we leave our cells everywhere we go. Those cells can travel to places we've never even been. So it's not impossible that your DNA could be found at a crime scene and cops could knock at your door. That's what happened in 2012 to a man named Lucas Anderson. He was in the hospital at the time of the murder. It turns out that the paramedics used the same oxygen monitor on his finger that they used on the murder victim's finger later that day. And that's how Lucas's DNA was transferred to the murder victim's fingernail. Lucas Anderson was in jail for 5 months before they figured out what had happened.